save that one there. All right, so what I want to do today, it should be a fairly quick one today. And what did I want to share? Share. Which one did I want to share? I don't want to share that. I don't know. They give you such tiny. Oh well, I mean that'll do. Okay, what are you doing here? All right, so this is the lab. It's going to be a bit weird, I have to say. I've never taught an online lab before, and in many ways, genetics lab isn't the best one to start with because a lot of the skills that we're trying to get you are hands-on skills, right? I mean, there is a whole swathe of genetics research which can be done only with a computer, particularly bioinformatics and comparative genomics, things like that. But there's an awful lot of genetics which you need to get your hands dirty, basically. So the aim for this is really to, how does it do that? God damn it. Try and give you as many of the skills that you would have gotten anyway, right? Particularly analytical skills. Pardon me. That's my lunch. That you really need later on, right? Even though we can't give you the hands-on skills, the hope is that we can give you enough of the kind of information and theory, and then tie that with some pretty robust analytical stuff. So you're going to develop like the thought processes, because at the end of the day, techniques are 10 a penny. Even when you end up in a research lab, doing a PhD, master's, working as a technician, or even working in a company doing, I don't know, whatever, pharmaceutical work, let's say, you'll be taught all the things that you need to know to be able to do your job, right? You're not going to be like left alone, like, hey, you go figure out how to do next gen sequencing. We're just leaving you to it. It doesn't really happen that way. So techniques are, I mean, it's good to know them, obviously, but really it's the conceptual basis or the analytical basis that matters. And so the aim, as best as I'm able to do so, is to give you that, right? Also, you'll find that the lab is, at least the Blackboard page, is far less populated than the lecture one. That's because I haven't done a lot of it yet, right? So I'm pretty much making this up as I go along. I've got a pretty good idea what I want to do. Haven't 100% figured out yet how I'm going to do it, but I will. Don't worry. It's all under control. So essentially, what we're going to do is a bunch of exercises, of which we're going to do one today, and in large part, those are going to be tied with Blackboard quizzes, essentially, right? So like little mini assignments. There's one already up for today's lab, which is basically getting you to do a bunch of the calculations which are in the lab handout, which is also posted, and then give me the answers, and then you get graded for that. You need to draw a graph at the end, right? So almost all of them are going to be a mix of those two kind of formats, where you're going to be asked to answer questions, but also upload images or files or whatever, which then I'll grade manually. There's that. We're going to do a little mini linkage project in the middle. If I could figure out how to share, let's see if I can find that one, wherever that's disappeared to. Yeah, where is it? For some reason, I don't have the genetics lab schedule available. Where you are at. All right, other applications. No. This is all a mystery to me, I have to say. That's not going to work. All right, well, anyway, if you open up the genetics lab schedule, then let's actually, sorry, I'm trying to figure out these controls. Share my meeting window. Can you see the genetics lab schedule? God knows what I just did there. No, that didn't work.
It was up for 1 second.
Yay!
Yeah,
Did you
I
catch
saw
everything
for
you
like
needed?
I saw a glimpse.
God damn it! All right. For some reason, I don't know why it's not. Let's get rid of this. Get rid of that. Maybe I have too many applications open. That's the issue. All right, let's try again. God damn it, this is such a pain in the backside. It lets me show my calculator, for example. Why would I want to share that with you lot? All right, well, let's just stick this. Oh no, now it's available. Let's see. Go on, it's thinking about it. There we go. Phew. Finally, that's what I was trying to show you all along. So, yeah, we've got a bunch of like the first, well, week, 2 weeks and a half, give or take, are a bunch of just generic kind of labs. Really, the idea here is to both expose you to some new stuff, right, in theory at least, and also give you some of those analytical skills that we're going to be using later. That are going to be really handy. And then what we're going to do is a little linkage project, right? And I think that ties in more or less with covering linkage in class. And that is the rump, I guess, of a semester-long fly project that we would normally do if we were doing this lab in person. And so the aim behind this is really to, one, kind of expose you to linkage in a real context, right? Not just a theoretical one, but also to get you developing some analytical skills in terms of data analysis and graphing, right? Which are really important for scientific work, and also doing a little mini lab report, right? Which will essentially take the graphs that you produce from the second lab of that series and stick that into a short but comprehensive lab report, basically saying, you know, what you would have done if you'd actually done that work and what you think it means, right? So kind of tying it together. Then after that, we'll be starting a large kind of linear experiment, right? Which is really like a molecular genetics analysis of phenotype and genotype. So we're going to be using a single phenotype from flies, the yellow phenotype, which is, as you would guess, makes them yellow. And then we're going to be working through a series of kind of online exercises where in silico, right, that's what they call it when you do stuff in your computer. It's like in vitro and in vivo in petri dish and in like real life animals. We're going to be doing this in silico. We're going to go through the steps of isolating the DNA from those flies, doing some quality control, doing some PCR, and then we're going to use later on some actual PCR sequences that we've generated over the last couple of years to look at the molecular basis of that phenotype. And in the process, you'll get some pretty in-depth exposure to some bioinformatics software, which is extraordinarily powerful. It's a bit of a pain in the ass to use because it's open source, and so it's written by a bunch of nerds who know this thing inside and out. but it's a very powerful piece of software, and you're going to be using that to um, essentially do what I or Dr. Page or any other genetics researchers would actually do if we were doing this for real, right? So this last half of the summer semester is very much a real life exercise that if you were in a molecular genetics lab, this is what you would be doing, right? This is exactly the same kind of stuff that I've done before. In fact, the first time I did this, more or less, was for my PhD many, many moons ago, right? So this is a pretty much a carbon copy of current modern genetic single gene analysis, right? So um, these are some really, really useful skills that will stand you in good stead if you ever end up going into that kind of work, right? And also it serves to show you like this is how you do this stuff, right? I mean, a lot of the labs that you do online or not tend to be kind of canned experiments in the way, and really in a sense that's what we're going to be doing for the first 2 and a bit weeks, right? They're really just like little standalone things to teach us some skills. Um, but really, what we're trying to get you to the level of at some point in the future, wherever that may be or wherever, is actually doing this stuff for real, right? Because that's how you're going to earn a wage, right? Whether that's a wage as a grad student, which is shit, or as a lab tech, which is way better, um, or hopefully even something even better than that. Right. So these are all kind of the skills that we want you to have because they're important. And we'll see how it goes, but hopefully you'll end up getting most of those skills anyway. You're not going to get the whole muscle memory of, pipetting out stuff into PCR tubes and, running it and then going, it didn't work, or yay, it worked, or whatever, right? You're not going to get that, unfortunately, because we just can't do that. But you will be getting the actual conceptual and analytical skills, right? That you would get if you were in person. So hopefully that's gonna get you at least 90% of what you would get anyway. As I said, I'm not making this up as I go along. That's probably a little bit harsh on myself. The modules 1 to 6 are all done. I haven't loaded them up yet because I want to do them in order, so it's a little bit easier to work through them. But these are already prepared. In fact, they're getting submitted to a journal in the next couple of months as a teaching research paper. So we've spent a lot of time thinking about these things. The first few ones, we're just going to put stuff together that's going to let you do what you need to do. And the aim is to get that at least the day before the lab. Ideally, I'll start, getting on top of this over the next couple of weeks. So in terms of, let's see if I can get someone else showing now. Pum, pum, pum. Where'd it go? In terms of assessment, come on. Basically, you're going to have, well, on average, 1 a week of these kind of online Blackboard assessments that you fill out. Some of those will be, as I said, just answering questions or doing calculations. Others, I'm actually going to need you to draw something and upload a copy of it, much as you would draw it on a lab handout, right? And because you can't give me lab handouts, this seems like the best way of doing so. And those will be kind of just filled out automatically. The deadline is the day of the next lab, usually midnight, just because that's the automatic default setting, and I'm kind of a little lazy to change it. So you're going to have a couple of days after every lab at least, right, depending on if it's Tuesday or Thursday, until you have to finish that off. But I would suggest getting those things done as soon as you can when they're fresh in your memory, because again, you're going to be constantly bombarded with stuff, right, that you need to do. So the better you stay on top of this stuff, the the better you'll do, essentially, and the more successful you'll be in this course. There's also going to be a couple of standalone things from the linkage lab series, right? So I'm going to give you a bunch of data and some instructions and videos and so on and so forth. And I'm going to get you to draw me a fancy graph, right? This isn't like one drawn in crayon with like smiley faces on it. This is going to be something that you would actually present to a journal, right? That kind of quality. And also the incorporating that into a lab report. So those together will be a fairly significant chunk of your grade, and I'll walk you through those in as much detail as you need prior to those being due, right? And that's pretty much it for the lab, to be honest. Go on. You need to have some more coffee, Angela. Can never have too much until you start pipetting stuff and you're like, I used to have to go easy on the espressos before I went in the lab. And that's pretty much it for the lab. So for today's lab, let's see if I can... Actually, you know what? I'm just going to draw it. Let's see if this is going to work. Funnily enough... Oh, yeah. That's kind of like intensive training there, that's for sure. Let's see if I can get up a picture of me. Let's stop sharing this. How do I stop sharing that? Oh, fuck knows. Am I recording this? I am. Good. I just want to get the picture of me back because I don't actually know what I'm pointing at. Oh, that's how that works. That's pretty cool. Sorry, discovering stuff as I go through. Let's stop that. And that should pop me back up. Yay, there I am. Cool. All right, so funnily enough, this is my personal whiteboard. I can't even see what I'm doing now. Let's put this over here a little bit. All right, so funnily enough, the last thing I drew on this for genetics was what I was trying to explain in terms of the template strand versus the strand of DNA that is encoding a gene, right? And so just because I don't want to rub it off before it's useful, this here is the coding strand, right? Remember I was talking about coding versus template? This here this strand, because there are two, because it's double helix, pardon me, this strand encodes the information, the gene, right? But because RNA can only be made from 5' to 3', right? And this is going from 5' to 3', you can't use the coding strand as a template. It's going in the wrong direction, right? You could only make RNA, if you only had the top strand to use, you could only make RNA going in the opposite direction to the direction of the gene. Does that make sense? Because it's anti-parallel, right? They always go in different directions, right? Just as you have 5' to 3' here, the bottom strand is 5' to 3', right? So if you want to use a strand of DNA as a template, right, remember that mRNA is going to be a direct copy of the coding strand. You're going to use the one underneath, because then if you have ATG as your first codon here in DNA, you're going to have the opposite down here. My arm isn't that long enough, right? And so the first codon in the mRNA is going to be AUG, right? And so that way, by using this as a template strand, you get to make the RNA in the right direction, right? It's from left to right, same as the gene goes from left to right. But also, you're going to end up with exactly the same letters, bar the whole uracil thymine deal, in the mRNA as you do in the template, as you do in the coding strand, right? Anyway, we'll come back to that in the future, but just because it was handily up there already, I think that was from like the last class that I had last semester. That's a neat way of explaining that. It's very hard to describe in words. All right, so let's get this off. I really need to get a better way of cleaning this because this looks like shit.
I've heard if you draw over the dry erase marker with a fresh dry erase marker, it comes off easily.
Oh yeah? Oh, that's cool. Thanks for the tip. I'll just get some rum. Actually, that would be a terrible waste of rum. I don't know. Get some nail varnish remover. All right. So essentially, what we're getting at today, right? Whatever you want to draw in terms of an axis, right? Here's your value, like whatever it is that you're measuring. Could be height, could be bushels of wheat, could be whatever you want. And here might be two different conditions. We're going to do height because it's an easy one, right? We just go around asking people. I'm going to go, you're up in the corner, go to floating panel view. What does that mean? Am I in the way?
No, that was from earlier whenever you were trying to figure out how to make yourself bigger.
Oh, right. Yeah, this is a work in progress, that's for sure. Right, so you could do height by lots of different things. By sex is probably easiest. Right, so essentially those are our two axes, right? We've got kind of categories at the bottom. This may be continuous, it may be discontinuous, it depends on the kind of data that you're looking at, right? And we have whatever value you're recording. My fan is kind of blowing off of the whiteboard and causing noise. So essentially, whenever you do this, right, you're going to be looking at a population, right? And in this case, our population would be the lab, right? The people that are present in the lab. Now, one thing that's really important in biology is that you rarely are ever able to measure every individual in a population. So typically, when you do any kind of survey or assessment or data collection or whatever, right? Even if it's going out into the hill country and collecting lizards, right? How many of this particular lizard do I find? Or even how many have this particular genetic makeup, right? Whatever you're going to be doing, you're going to be looking at a subset of the population, right? Because maybe we could get the height of everybody in genetics lab, but you're going to be a subset of the Texas A&M San Antonio population, right? Which is in turn a subset of the San Antonio population and so on and so forth, right? We're not going to be able to get data from everyone. And so we need to have ways of turning these data, right? And so essentially, you're going to have stuff like that, right? You're going to have a range because not everybody's different, and that's why you're collecting the data. It'd be really boring otherwise. And then you have something like so for males, let's say, right? So now what we need to do is we need to figure out, okay, what do all those different points mean, right? How varied are they? How well do they represent the broader population? And so this comes into statistics, right? Everyone's like, oh my God, statistics are so boring. And, sometimes I would agree with you. Statistics is pretty boring, but it's also really, really useful. And so we have two kinds of statistics that we care about, or at least like this is Matt Crook's version. One is descriptive, right? So if you have two different populations, well, are they the same, right? What's the average height of females and males? How much does that population that you surveyed, how much does it vary from that average? Right, that's really more talking about variance or standard deviation, right? So how big of a difference do you have within each population? But also once you have that, because that's all good and interesting on its own, now what you're going to the questions you want to start asking are, well, are these two different? Are these two representative of the broader population? Or did we just happen to have like an entire side and backup team of basketball players in the lab, right? Which are going to completely skew your data set, typically towards the upper end, right? And so typically whenever you do really any kind of genetic work, any kind of biological work, you start with a lot of different data points, and then you got to boil it down to more digestible figures. And that's the same whether you're doing molecular genetics or if you're counting lizards or giant fluffy pandas or whatever, right? It's the same principle, because it's very hard to look at these two things and go, are they different? Yeah, maybe, maybe not, right? As humans, we don't tend to work very well with multiple pieces of information. We tend to try and narrow it down to as few bits of information as possible and then make a judgment based on that, right? And so that's really what we're going to be doing today during lab. And I'm going to take you through the principles. And then when you go onto Blackboard for the lab, you go to course content, I think. There should be an assignment there. Click on that. And that will basically take you through, boom, boom, boom. Let's see. I got to go through the whole sharing thing again. That. Come on. That will take you through this, right, which is a lab handout for today's lab. And basically, here's all the blurb, right, background and kind of the stuff that I'm giving you right now, to be honest. And these really will describe how to do what you need to do. Then when you go down here, okay, we're going to figure out how tall you are, right? Because that's going to be difficult to collate online. But essentially what we're going to do is we're going to start question 2 in Blackboard, and you're going to go through and you're going to calculate all of these for me. The vectors, the data collections are in the questions on Blackboard, right? So you're going to take those data and then you're going to calculate the mean, for example, for the males. Enter that, hit return, right? So there is a little bit of wiggle room, but do follow the directions. So if it says 2 decimal places, try and put 2 decimal places, right? So it's going to be, I don't know, 161.00, let's say, right? And so essentially the assessment is going to be doing all of this. And then at the very end, you're going to draw me a graph. And you could do it in crayon if you want, right? But you're going to have to take a picture of it, right? As long as I can read it, like, and figure out what exactly you've done, you're going to recapitulate this and then upload that to that assignment. Then I'll look at that and go, yep, that's good. And then you can get your marks. So essentially let's stop doing this. What we care about are a couple of different things, right? We care about differences between populations and differences within populations. And so I'll show you, well, let's do 2, right? Don't know if there's the same number of dots or not. I didn't count the original ones. But if you look at these 2, when you calculate an average, your average is denoted by that straight line are going to be the same. Are those 2 populations the same? That would be a negative, right? They are not. One of those populations of females that were being measured for height is very homogeneous. I would actually say those are probably female clone troopers from Star Wars, right? Or maybe we got a family of sex tuplets or something in the lab and they're all exactly the same. It's possible, who knows? Whereas this other population is very much more varied. Yet, when you take these 2 pools of data and you calculate the average, don't tell me I need a new pen. That's what you get. How can you tell if they're different now? Right? You can't. So what this lab today is about is a couple of different things. What does this population look like? How can we describe it, right? In as few terms as possible, right? And so what we're going to use is average. Actually, I think that's supposed to be like a X, something like that. I don't know. I can't remember. I'm not much of a statistician, right? That's the bar, right? That's the, it's not the middle one, right? Because that's the median, but it's the, basically add them all together and divide it by how many you have. That gives you the average, right? This is all simple stuff. You know this stuff already, right? Sometimes we want the median because if we have population that looks like that, where everybody's the same and you've got this weird outlier up here, the median, the average for this population is going to end up somewhere up here, right? Which doesn't truly reflect what the population looks like because of this weird ass outlier. Maybe we've got like some Guinness Book of Records height person that's visiting for like an exchange visit or something. But anyway, so sometimes you want to use median, particularly if the data are not normally distributed, right? So normal distribution looks like this, and your average is kind of more or less where the peak is, right? If you've got a really weird ass distribution like this, right, the average may end up somewhere different from where it really should be, right, where most of those people are, right? So that's why we use median. A median is literally the middle one, right? So you line up all of the individuals in your data set in a row from high to low, and you get the one that's in the middle. And what that does is it gets rid of those weird extreme outliers, right? So more, in the case of non-normal distribution, it's a more accurate term. But typically we use an average or a mean. Then what we want to know is how varied are these individuals from the mean? And so what we're going to calculate now, and this is a little bit of a trippy kind of thing to think about, we're going to calculate for each one of these points, what is the difference between that point and the mean? So for this point, there, we're going to subtract this value from this, the average from that value, right? So it's going to be x minus the mean. That gives us the difference, right? But the problem with doing that is that when you go down here, you're going to get negative values, right? So we employ a trick because we're tricky. Never trust statisticians, dodgy bunch, right? We're going to square that, right? Because if you square a number, even if it's a negative number, it will come out positive, right? So that deals with the issue of negative numbers. It causes other problems too, right? Now, we need to take into account how many individuals we have, because the more individuals you have, the more confident you are of your mean being a true representation of the population, right? Does that make sense? It's like if you measure two people, you get an average, right, which is like half a person. But depending on who you picked, even by random, you could end up picking like the two tallest people or two shortest people in the class. So the more people you survey, the more confident you are that your average is actually a true representation of the population. And so we need to take into account numbers, sample size. And for reasons I can actually never remember, I keep saying this and I should just go and look it up. We use that as the sample size, which is always n minus 1. And then we add all of these. You'll see this when it comes to chi-square. This means sum. So you add all of those differences together and then you divide them by n minus 1. Right, so in the case of the data that's on Blackboard for you, you've got 7 different heights. you'll be dividing by 7 minus 1, which would equal 6. Right? Does that make sense? And I don't know why it's like there are statistical reasons for that. I just can't remember what they are. Because as you probably guessed, I don't read statistic textbooks for fun at night. Okay, so what this gets you is variance. This is going to be kind of weird with me peeking over the top like this. But the issue that you get here, so that's a really useful measure of the variety, let's say, in the population. The problems with this is, let's use a different color, just because I can. The squared part. So in order to get everything with a positive value, so that we don't end up subtracting negative values from positive values and getting a weird result, we have to square stuff. But because we square things, now your units are also squared. So instead of dealing with height, we're now actually dealing with area. And that's not what we're measuring at all, right? So now we do something real simple, and we take the square root of that. Come on, why don't you let me go over there? Ah. There we go. And so what that gets you is it gets rid of the whole squared nonsense. And now we get back to, there's got to be more comfortable places than doing this one on the couch, right? That gets a standard deviation. Or SD. Ta-da! And so that's a much more useful and commonly used measure of variance in a population. And so if you were to look at these two populations now, right, we take away, essentially you take away all the data, right, because you're now converting it into a mean, like which is one single value, right? So now we have our two means still, but now we're going to add standard deviation, and we're going to have variation. Actually, let's make these a little bit bigger. It doesn't necessarily reflect all of the variation, right? You know, the most extremes in a population. But what it does is give you a much better sense of variation. And now when you look at the second population of women whose height we collected, now you can see that they're obviously very different. Even though we've taken away all of the data and converted it into a single figure, which is the mean, now we have a much more useful visual representation of how varied those populations are that contributed to the mean. And so another reason for doing this too is that if you were to, let's put the lid on this so it doesn't dry out. Let's say you did this again next semester. You know, I'm teaching this again in the fall, and I've got 2 lab sections. So I'll be doing this with 2 lab sections. Now what you can do is say, okay, if this is the range, right? This is the variance, or how much you would expect the individuals in a population to vary in terms of height. What you can now say is that, light reflected from my room light, when you do this again, you will expect with a high degree of confidence that when you repeat this, the mean will end up somewhere between these 2 upper and lower limits. right? Essentially, it gives you a confidence that even if you did screw up somehow in your data collection or there was some like weirdness going on, if you were to repeat this whole process, then the second and third and fourth or fifth, whatever time you did this again, your mean should be more or less, it's not going to be down here, right? It's not going to be up here. As long as you used a somewhat random like group of people, that should tell you that your mean is going to be somewhere within here, right? And so this essentially gives you the upper and lower bounds of the population, the bigger population, right? If we did it for the whole of Texas A&M San Antonio, you would expect the mean would be somewhere in here. It's not going to be the same mean just because you measured different people, but it's going to be somewhere between these upper and lower bounds, right? That's one way in which standard deviations are really, really useful. And that also means that if you had, and we can do this again for males, let's kind of go over here, right? So here's, a large random population. These are both have variance. The mean's a little bit higher than females, because often that's the case. But by and large, they're similar populations, right? They don't really vary hugely in terms of height. Now, if you look at these second populations, this one here and this one here, the means are different, but also the error bars, the standard deviations don't really overlap. And so what that means statistically is doesn't matter how many times you repeat this experiment, you're always going to see this difference from this one. So now you can start making conclusions and saying, hang on, gents are always taller than ladies. Why is that the case? Is there something going on in terms of genetics? Is there something going on in terms of environment? Why is there this difference? Whereas if you got these data, you'd be like, well, there is some small difference, but there's nothing enough that we can really tease it out and be confident of stating it. So now we've gone from this collection of data points, this kind of cloud of data, kind of move that, there we go, to being able to quite easily and visually say, yeah, these are more or less the same, or no, these are pretty different. You can see how much easier that is to kind of visually grasp, right? Than it was the previous series of dots, or even worse, like lists of numbers, right? So this is the kind of process that whenever you do any kind of scientific work, really, or biological work for sure, you're going to collect huge reams of data. I've got Excel spreadsheets with thousands of data points in, right? And I don't even do like big data stuff, typically speaking, right? But you have to boil that down into a form that people can understand, right? And this is part of that process. Now, the final thing that is really handy, largely because it makes these error bars look smaller, not that we want to manipulate statistics or anything, is something called standard error of the mean. Now, that's really easy. So the standard error of the mean is standard deviation, SD, and then you divide that by the square root of the sample size. And so basically what that does is when you have a really small sample size, your standard error of the means is still quite large. But what it means is that the more you, let's say, instead of just data from one genetics lab, we did data from, I don't know, 4 or 5. So then when you divide the standard deviations that we collect from all of those, but now you divide them by the square root of 4, which is 2, your confidence narrows, right? So you're now able to say, yeah, I'm really, really sure, rather than just being this. Now I'm even more confident, right? You kind of go from here to here because now we know we've done enough sampling that, you know, we're really much more confident about where we're going to find our mean. So that's called standard error of the mean. And so what all you're going to do today is work through and calculate those. Right, so I could give you, no, I don't know, I'd have to teach you a whole bunch of Excel. So you can do this in Excel, that's what I do. But what I'd recommend doing, because Excel doesn't do variance very well, is actually calculate the variance yourself by hand. It's a little tedious, but you only have 7 data points. So let's get rid of this. Anybody got any questions so far before I trundle on? No. And to be honest, after you do this enough, it just becomes second nature, right? You don't even really think about it. And to be honest, I never do this by hand because it's a real butt ache, basically. I use Excel because it's so fast. So essentially, for variance, that's going to be, remember again, the sum of x, and that's often like with a number, like x1, x2. Let's do it this way, that's easier. Divided by the mean squared, right? And so sum means basically you're going to have the second data point. There we go. Minus the mean squared plus third data point minus the mean squared, and so on and so on and so on for however many data points you have, right? And then all of that is divided by n minus 1. That's the variance, right? So that's the one that you really do need to do by hand. You can do the other ones in Excel, and I've got no issues with you. Sorry, I don't mean to mute myself. I've got no issues with you doing that because that's how everybody does it. If you want to know, let's see. Let's see. Hang on, I'm just gonna. Pardon me. All right, let's see if I can share this with you. Oh, yeah. Excel is the bomb. It's a real pain in the backside for drawing graphs because it seems to think it knows better than you do what kind of graph you want to draw. But for doing any kind of data analysis, it is marvelous. Let's see if I can open that up for you. Yeah, there we go. And also, I've got absolutely no desire for you to spend hours on this. I'd much rather you learn how to do it quicker and more efficiently. Okay, so can you all see the Excel spreadsheet? Yep. Okay, so whenever you want to calculate a function in Excel, you put equals in because that tells Excel that you're going to start putting in a formula. So it could be, you know, something as simple as this plus this, right? Equals 5. Yeah, that's not very interesting. But whenever you put the equal sign in, it tells Excel you're going to be typing in some kind of formula. And it'll have a complete fit if you don't, because it's expecting that. So different versions of Excel vary because of, you know, Microsoft needs to change things in order to justify charging people for it. But in the version I have, and you'll probably find this somewhere on your version, even on like Google Numbers, whatever it's called, like the free Google version, go up here and select average, right? That will bring up a box. And what this box is basically defining the range, like what numbers do you want to use. It does a pretty good job of guessing, but if you do another one beneath this, it will try and include that one too, right? So you can change this. So you can click on that and you can change it manually. I'm just going to hit enter. Or if you click on it here, you can actually see the kind of data that's kind of within that range, right? So you can actually, if you carefully, right, you see that little double-headed arrow? You can just drag that down, right? Hit return. Now the average is going to be calculated from a different subset of the data. Right, oh, and Control Z, wonderful. I'll just get you back to whatever you did before. Right, so quite often what I do is put like little markers on the side so I can remember what I'm doing. Median, it's tempting to get you to do this manually just because that's a little bit sadistic in a sense, but it's so much easier to do it here. Do the same, but now you got, oh yes, lab lectures, I'm recording them too. They'll also go up on the YouTube channel. I'll be doing that after this, and I'll send you the link as soon as I've done so you can have a look at them. All right, so you can go back and like, what the hell is he talking about? So this also shows you that kind of little hiccup, because again, Excel thinks it knows better than you do what you want to do. Sometimes it is right as well. So you can either just change that manually, right? Go like so, or let's do it again. Let's hit return, and now we can actually pick up this corner. Come on. It's been a little bit fiddly, unfortunately. Yeah, let's just do it the other way, a lot better. Sometimes it's hard to pick up those corners because they don't, they overlap with other things. So in Excel, you can have access to all these functions. Now, as again, I said, don't use Excel to calculate variance. I tried doing it and it gave me the wrong result. You're going to have to do this manually. However, what you can do, here we go. Excel fun. Who does not enjoy Excel? Don't put your hands up. All right, so you can get all kinds of crazy if you wish to. What I'm going to do now is I'm going to calculate the variance for each of those points. Right, so shout out if I lose you, right? Please. Just go, what the hell are you talking about? Okay, again, equals. Because that's telling Excel you're going to put a formula, not just a data point. Now what I want to do is subtract the average from this data point. So I'm going to do that minus that, right? Crab sticks, I've got a negative value. I don't want that because when I add it together in the sum, it's going to take away from the positive values. So let's go back to this. Now in Excel, when you want functions being done to a specific thing, you have to kind of protect those things with a set of brackets. So if we put in brackets here, whatever function we do after that is going to apply to, oh, there you go, absolute value. Yeah, you see, you know more about this stuff than I do. This is just like what I've learned off the back of a packet of cereal kind of thing. Now you can do an uppercase, and that will square it. So again, let's go back here. So you don't want to do that without the brackets or whatever Andrew was talking about, because then it will square the last value in that series, and you don't want that. You want it to square the absolute value. There you go, thanks. Now what you can do, because Excel is also super cool, I'm a little bit of a fan, as you probably guessed. You see there's that little square on the bottom right? Grab that and drag it down. Now, The only problem with that is that it's going to keep that relative position the same all the way down, which is not what you want. And so you often just have to go through this manually and just change all of these to that. You can also just grab the square as well if you fancy that. That's totally fine. You can do it manually. That's just the downside. But one thing that you should be aware of is that if you do multiple lots, like you're going to have male and female, all you need to do is maintain the correct spacing between the two, and you can copy this entire thing over to the male, the whatever the other data set is. And do. Okay, so now these are all of the variances. Now this is not the final version, right? Because that's just the individual data points. And so if you remember, right, variance is the sum of all of these, right? So we've got to add all of these together, and then we have to minus sample size minus 1. Now you can actually do a count function as well. I'm not going to do that because that's unnecessarily complicated. But if you want variance, stick it in over here. Okay, what we're going to have now is, now we can sum things. And again, if I'm going too fast or you miss something, just shout out and I'll go back and show it to you, okay? Click sum, and that's going to ask you, well, what do you want summing, okay? And I don't want those things summing. So put your cursor over this one and just drag it. Right, and now you see it's sum D1 colon D8. So that gives you the range like so, right? So that's the sum of the variances, but it's not variance because we need to divide by the sample size minus 1. So now we're going to put brackets around these, right? Remember, this is a distinct function. So we want to kind of wall that off from everything else. And I'm just going to do this manually because I know that there's 8, so it's divided by 7. You could actually use a count function and then you could do it as a formula too if you wished. But I think I'd probably, kind of blow your brains out if I try to do all of that at once. So that's what, 7? There you go. That's it. Right? Always, actually, which, is that a parentheses or is that parentheses? I'm not much of a grammatician either. Oh, okay, yeah, parentheses then.
Or brackets? No, not brackets. Those are more square.
Yeah, sorry. Kind of Excel learn on the streets kind of thing here, or English learn on the streets. Actually English as well. I should know this stuff better. Yeah, they're parentheses. Not entirely sure what you use the brackets for, but anyway, yeah, they should look like that, not the square ones. You could try it with the other ones, see if it makes any difference. I don't know whether it would or it wouldn't. And then you can just simply work out the other stuff, right? And so this is, you could either do, crap, I'm showing you how to do all of this stuff. Where's the fun in that, right? So standard deviation, you could either do it directly by doing that, right? So you use the equals stdev function and then select the data that you want to find the standard deviation of, right? Or, let's see if I can remember how to do this. I can't remember it off the top of my head, so there we go. Skirt. That's what we want. We want skirt. And in there, we want that number. Should, hopefully, let's compare them, give you the same answer. I'll be really embarrassed if they don't. Shit bits, they don't. Okay. let's forget that one then. I'll have to figure that one out myself. And then the standard error of the mean is simply that divided by the square root of the sample size, which is 8. I should have to have a look into that because the variance was coming up differently too. But in this case, and again, I'm really not quite sure what's going on there. I'll have to talk with a friend of mine. Follow the steps in the lab handout, right, which is calculating the variance individually for each point, and then using this sum of the variances minus n minus 1. Oh, hang on. That's why, dumbass. There you go. Oh, honestly, where do you find these people? Okay, so obviously be careful what, symbols you put in there. Phew. Okay, I've got to go back and double check, make sure I actually did the stuff for the assignment correctly. Yeah, there we go. It's the same thing. Phew. I will go back and check because that would be really embarrassing otherwise. And then the SEM is just that. I don't think there's a function for doing that directly in Excel that I've found. And so if you want to get all fancy, that's going to be that. How come none of you biostats people pulled me up on that? Honestly, that was a test. There you go. See whether you're paying attention or not. So we want to square root. What's that? 8. Let's see if this works. Hey, it did work. That's how you get the standard error of the mean. Right, so essentially that's what I'm going to be getting you to do today. And then what I want you to do is take this average for both female and male heights separately, obviously. And I want you to plot that, and it could be done in Excel if you're handy with Excel. You can draw it out and take a picture of it with your phone. I really don't mind. You could hand draw it, whatever. You could use like Adobe Photoshop if you want to be super fancy. And then I want you to plot error bars on that, which are the standard deviation. So +1 standard deviation, -1 standard deviation for both the male and the female heights, averages or means, right? Do that, post it up onto the Blackboard thing, and you'll be all good, essentially. And that's pretty much it for today, right? So it's really kind of just giving you some handy dandy skills, showing you a little bit of Excel wizardry. And honestly, this is stuff that I do all the time for my research, right? To take big lots of data and kind of get it into a manageable form. And also just to get you familiar with working with data, right? Because you're going to be doing that a bunch of times later on in the semester. And really, it just needs to become second nature. You're just going to do this without even really thinking about it. All right. Does anybody have any questions about anything so far? Pardon me. And I promise you, right after this, before I go make myself a coffee, I'll go and double check my sums for the assignment. Because that would probably explain why it came out so different last time. Anyway. Cool. All right, so that was a pretty quick one today. Generally speaking, a lot of these labs will be pretty quick because obviously we're not going to be doing physically any of the work, right? We're not going to be running gels and stuff like that. So I've got these pegged out for like the full lab period, but most likely we're not going to use any of that. Peyton, for sure, I'll send you a separate link, okay? And that's pretty much it. All right. As soon as this recording is converted, I'll post it up onto YouTube along with the lecture from this morning, and I'll share the playlist of that as well. All right. See you all on Thursday. If you have any questions between now and then, just send me an email and I'll get to them as soon as I can.
Thank you.
Oh, quick question from Crystal Linda. The heights will be in the questions on Blackboard. So each of the questions that you see in Blackboard, don't do them yet, because again, I need to double check the math. Each of the questions will have all of the data points you need to calculate that figure. But really, in terms of, just a second, Austin, in terms of doing this efficiently, try and do an Excel. You can do it by hand as well. That's totally fine too. But, I'm not teaching you to use an abacus or, write with a pencil here. I'm trying to give you the skills you'll actually use. So use Excel if you can. Question from Austin about when this is due. This will be due Thursday evening at midnight. So if you do have any questions about this that you can't figure out either on your own or by email, you can ask me next lab session too. All right. Awesome sauce. Cool. Thanks very much, everybody. Enjoy your day tomorrow and see you tomorrow, Thursday.